glucose molecule, right? Remember, glycogen is just glucose that's stored, right? It's linked. So when we say we're breaking down glycogen, we're literally breaking off glucose units off this larger glycogen molecule, right? So what glycogen phosphorylase does is it takes an inorganic phosphate that's just floating around the cell, grabs it, puts it onto the glucose molecule, and now we have glucose 1-phosphate, okay? The only difference between this molecule here, glucose 6-phosphate, and glucose 1-phosphate is just the location of the phosphate on that carbohydrate, right? And the glucose 1-phosphate, that phosphate's on the 1 carbon. On glucose 6-phosphate, it's on the 6, right? So still a 6-carbon molecule, still has 1 phosphate on it. The only thing that differs is the location of that phosphate. But why is that important? It's important because this molecule, right, PFK, is not going to work with, it's going to work with fructose 6-phosphate, but this enzyme here, glucose phosphate isomerase moves the molecule around, moves carbons around, moves atoms around, right? This is not going to be able to work with an enzyme that has a phosphate on the 1 carbon. It only works with an enzyme that has a phosphate on the 6 carbon, right? So when we start with glycogen to make ATP, right, there's an additional step. We have to move that phosphate from the 1 carbon to the 6 carbon, right? And now we have G6P, okay? So there's no use of ATP here, but there is an extra step, right? It's kind of the trade-off. In any case, we end up with glucose 6-phosphate. This is the starting molecule of glycolysis. Once we have that, we can proceed to try to make energy out of it. Okay? This makes sense? So if we learn in 346, why is there a net difference between ATP production between glucose or glycogen? Right? The difference comes from the fact that when you start with glucose, there's an additional priming reaction. You have to use an ATP to add the phosphate to glucose, and there's another ATP used in the PFK reaction. But when you start with glycogen, this enzyme does not use that ATP. It just uses a phosphate from the cytoplasm. So that's where the net difference comes from. Does that make sense? Okay, so I'm circling that, and I told you that, I'm telling you that because on the exam, students confuse what hexokinase does and what phosphorylase does, right? You can understand the process of, when you break down glycogen, it doesn't use an ATP because the phosphate comes from the cytoplasm. If you understand that concept, that's great, right? But if you're calling that enzyme hexokinase, on the exam, you're going to get all of those questions wrong because what you think is hexokinase, what you think is phosphorylase, you're calling hexokinase, which actually does use an ATP, right? So make sure you know these terms. These enzymes go with specific substrates. They work in specific steps, okay? So don't say, if on the exam they say, the breakdown of glycogen involves which of these enzymes, and you are choosing hexokinase, that's incorrect. It doesn't work with that, right? It's glycogen phosphorylase, and then vice versa. What enzyme traps glucose into the cell upon entering? It would be hexokinase, right? It doesn't use an ATP, true. Right? So that's the important part there. So once we have glucose 6-phosphate, we literally just move that carbon molecule on the carbohydrate around. So fructose and glucose are still a 6-carbon molecule, just the structures are slightly different, right? So if we look at what's basically on this molecule, you have a 6-carbon carbohydrate with 1 phosphate. We slightly change the structure of glucose into fructose. It's still a 6-carbon molecule, still has a phosphate on the 6-carbon, and now this fructose 6-phosphate can then be phosphorylated by PFK. Okay, what does PFK do? It uses an ATP to add another phosphate to fructose. Notice how the names of these substrates changes as they go through being interacted with the enzymes, right? Glycogen phosphorylase adds a phosphate. So now we go from glucose, glycogen, to glucose 1-phosphate. Hexokinase adds a phosphate to glucose. Now it's glucose 6-phosphate, right? So the structure, the name is telling you the structure and also where that phosphate is located. Okay. Fructose 6-phosphate, it's a carbohydrate with a phosphate on the 6-carbon. Then what does PFK do? It adds another phosphate. Now it's fructose 1,6-bisphosphate, right? Bi, bis, 2, okay? Where are the phosphates? They're on the 1 carbon and they're on the 6 carbon. It's in the name. Okay, so this, you can literally see in the name, like what does PFK do? It takes a phosphate off of ATP, puts it onto that fructose molecule, and fructose 1,6-bisphosphate. From there, this is a 6-carbon molecule, right? We're going to split it in half, and it will become a 3-carbon molecule and a 3-carbon molecule with a phosphate on each of them. Okay. These steps in glycolysis, this is where we take the phosphates off of this substrate and resynthesize ATP. Okay. This is really important because up until this point, we've just invested to ATP. Right? We haven't actually made the ATP until after this NAD plus dependent step. Okay? After this NAD plus dependent step, then we're able to make ATP here, make ATP here. Remember, this happens on both sides, it happens twice, so that's where the production is net positive 2. Right? We produce 4 ATP, but net is actually 2 for sugar and glucose. Okay? Remember this from 346? So on the exam, I'm not going to ask you specifically, like, what is 3-phosphoglycerate? You'll never have to draw out every step of glycolysis. You don't even have to memorize every enzyme in glycolysis. But there are key steps in glycolysis that are important in driving metabolism to increase so we can generate more ATP. Okay, so it's understanding the concept of these specific steps of glycolysis that we're going to try to focus in on. So this third step of glycolysis is the PFK reaction. It's the rate-limiting enzyme of glycolysis. Okay, what does PFK do? I mentioned we have fructose 6-phosphate. Okay, we have an ATP. We have PFK, and essentially it's just taking a phosphate off of the ATP and putting it onto the fructose molecule. Right. So now we have 6 carbons, a phosphate apiece. Okay, we have an ADP because that's what's left after breaking down ATP. And from there, then we will split that molecule into 2. Okay? This process is irreversible. Okay, so why is it important? Because if we think about it, in here in glycolysis, if we bring glucose into the cell and we use an ATP to phosphorylate the glucose and keep it here, we could use glucose 6-phosphate in glycolysis. But what if we don't need to use that glucose 6-phosphate? What if we already have a bunch of ATP? That glucose is now brought into the cell, but what do we do with it then? What can we do with it?
Glycogen.
We can store it as glycogen, right? So when we bring glucose into the cell, we have to invest in ATP to keep it there. But once it's in the cell and it's phosphorylated, we can actually store that as glycogen so that the next time we need to utilize that glucose molecule, it doesn't cost us an ATP, right? Imagine if this glycogen breakdown required us to use another ATP. Well, then we would have used ATP to bring it in the cell. We would have used another ATP to actually break it down in the stored form. But the benefit is that once we bring it into the cell, we've already invested that ATP. So we either use it in glycolysis to make more ATP, or we store it as glycogen, and when we need to access that glycogen, it doesn't cost us another ATP because we've already invested it in the first step to even get it in the cell. Okay? So the investment in ATP here is maybe not so consequential, but if we think about this one, right, we could use another ATP down here, but if conditions are not prime, then essentially we've broken down glucose, we've changed the form into fructose, which we're not going to be able to store, okay, and now we have some phosphates on it. So imagine if there were not optimal conditions to produce ATP down here, we essentially would have invested another ATP down here for no reason. Okay. The fructose is supposed to be metabolized and go through glycolysis, but we're not going to use that directly yet if there's not conditions to satisfy glycolysis. Right? You could use it, but it depends on the needs of glycolysis. So you would have broken down the glucose without really any need to do anything with the rest of it.
So would it just stay there?
It could just stay there, right? It could be converted into other forms, but it wouldn't be necessarily a form to generate ATP immediately unless there was a need for the ATP in glycolysis. So one key part I want to talk about here in glycolysis that I'll mention in the next few slides is this NAD plus dependent step. Just a reminder, what does NAD plus do in metabolism?
Substitution.
Yeah, it carries hydrogen ions, okay? So when it has a hydrogen on it, right, it's NADH. These NADHs, where do they go in metabolism? They go to the electron transport chain, okay? They get to the electron transport chain and then they drop off their hydrogens, right, into the inner membrane space, and in that process you can make ATP at the end of the electron transport chain with oxygen. Okay? So the whole idea around them dropping off the hydrogens is if there's enough oxygen in the electron transport chain to be able to drop it off and make water and ATP. If there's not enough oxygen, then this NADH essentially just holds on to the hydrogen, okay? So if you have NADH that has to hold on to a hydrogen, what about NAD+? How do you get NAD+? You can't unless you take that hydrogen off, right? So think about it, like this step in glycolysis requires NAD+, okay? But imagine if metabolism, you already have a bunch of hydrogen ions floating around, there's a lot of hydrolysis going on, so all these NAD pluses are now NADHs because they're holding on to hydrogens, okay? This step in glycolysis requires NAD+, right? If you don't have any NAD+, you can't make this ATP and you can't make this ATP. Right? So what ends up happening is that in glycolysis, when there's not enough oxygen available, or if there's not enough oxygen available, then in all likelihood you probably don't have enough NAD+, right? Because NADH drops off to the electron transport chain. So if you don't have enough NAD+, what happens is that these hydrogens can come off of NADH and they get put onto pyruvate, and that's how we make lactate. Okay, so because of that, I'm just kind of preemptively kind of introducing this because this step is really important, because once we get to this step, now we can make ATP, a positive net gain, right? But up until this point, all we've done is invested to ATP. Right? So whether there's NAD+ available or not is going to dictate how we're going to use these substrates to make ATP, right? If there's not enough NAD+, then it's going to make it into lactate, so then we do have an NAD+, and if there's enough NAD+, then it just goes to pyruvate and everything gets oxidized and oxidative, okay? But this is really key because essentially if we weren't able to move these hydrogens around off of NADH, we would essentially just stop contracting because there'd be no other way to make ATP anaerobically. And then just a reminder, the difference between substrate level phosphorylation and oxidative phosphorylation. Oxidative phosphorylation is where you're using oxygen essentially in the electron transport chain with ATP synthase to synthesize ATP, right? So the presence of oxygen causes the phosphorylation of ADP, oxidative phosphorylation. So we're phosphorylating ADP because oxygen is present. Substrate level phosphorylation is literally taking phosphates off of one substrate and putting them onto ADP. Prime example is this right here. We have 1,3-diphosphoglycerate. We take a phosphate off of 1,3-diphosphoglycerate, we put it onto ADP, now we've synthesized ATP. And now this substrate has one less phosphate on it, right? So this is substrate level phosphorylation, right? We're phosphorylating ATP, we're resynthesizing ATP, but it's because we're taking phosphates off of substrates. It has nothing to do with oxygen being available for phosphorylation. Yes?
So, if we don't have any NAD+, we just have an NADH, does pyruvate get converted into lactate?
Yes. Yeah. And I'll show that in the next few slides, right? So that process allows us to use carbohydrates anaerobically to make ATP, which is different than fats, right? Fats, there's no way to use fats anaerobically. Every step in, fats being available to use for ATP production depends on the presence of oxygen. But because lactate dehydrogenase can form lactate, it allows us to use carbohydrates anaerobically, which is why it's the only substrate we can use during high intensity activity or anaerobic, because we have that ability to do that with carbs, but not fats. Right. So that third reaction of glycolysis is highly regulated. Okay, it's an irreversible step. Essentially, you want to have enough signals or sign in metabolism to make sure that you're going to be able to produce ATP in glycolysis. So the regulation of PFK is really tightly regulated. Okay, so ATP is an allosteric inhibitor and AMP is an allosteric activator. What I mean by that is that ATP will inhibit the activity of PFK. If you have a lot of AMP, that can override the inhibition of ATP. Okay, so ATP will slow down the activity of PFK. AMP will increase the activity of PFK. And citrate is also an allosteric inhibitor. So when we think about it, all of these bioenergetic pathways, they all increase their activity to supply more ATP, right? So if your cell has a low amount of ATP in it, is the energy status of the cell high or low?
No.
Low, right? ATP is energy, right? It's what we use for energy. So if we don't have enough ATP, then the energy status of the cell is low. These bioenergetic pathways, they sense that low energy status, so then they turn on so we can make more ATP and bring our energy status back up to what is homeostasis. So think about it. If PFK is a rate-limiting enzyme of glycolysis, it's because it's going to increase glycolysis to make more ATP if there's not enough energy in the cell. In all circumstances where there's already a lot of energy, glycolysis does not need to be turned on very high, because the point of that process is to make even more energy. Well, why do you care if you already have enough of it, right? So this explains why ATP inhibits that enzyme, which slows down that pathway. How do you get AMP? Where does AMP come from?
So like
Yeah, so first you have to break down an ATP, right? Then you're left with an ADP. And then you can break down that ADP even more into AMP. Or you could take two ADP, put them together, you have an ATP and then two AMP. Right? So the idea is that you can, when you have more AMP, it's essentially a signal that we're using more ATP. Okay, more ATP breakdown. So the more we break down ATP, the more this inhibition is removed, and the more the activation is increased, because they're essentially two sides of the coin, right? So if we look at what regulates PFK, there's 5 allosteric regulators. ATP inhibits PFK activity, AMP and ADP increase. Fructose 2,6-bisphosphate increases. You don't need to remember that because we don't really cover it a lot in this class. And then citrate also inhibits. Okay, where is citrate located? In the Krebs cycle, right? So citrate is a aerobic substrate, right? So it's located in the Krebs cycle. If you have a lot of citrate, okay, it's really probable that we're not using that citrate in the Krebs cycle, right? Because when you have a substrate and there's a need for it, it gets converted into the next product, right? It gets used right away. So when you make citrate at the start of the Krebs cycle, if that's building up and it's not being used, it actually feeds back into PFK to slow down its activation, right? Because where does, actually, back up, to remind you from 346, where does citrate come from? How do we make citrate? It's in the Krebs cycle. It's the first product of this Krebs cycle, right? But what actually makes up citrate synthase? A citrate. One is the enzyme citrate synthase, right? But what does citrate synthase use to make citrate? Acetyl-CoA and it's a cycle, right? So oxaloacetate, right? Oxaloacetate is the last product of the Krebs cycle. So that last product of the Krebs cycle, oxaloacetate, joins with acetyl-CoA. With citrate synthase, they come together and that makes citrate. So that first step of citrate production, if there is a need for the Krebs cycle to be highly elevated, that citrate is going to be used right away. But if there's not a high need for that citrate, then it starts to build up. right? That acetyl-CoA, that first made citrate, that came from a pyruvate, right? Isn't pyruvate converted into acetyl-CoA?
Yeah.
Yeah, right? So imagine you have glycolysis that's really active. It's producing a bunch of pyruvates, right? Those pyruvates want to be converted into acetyl-CoA. They go to the Krebs cycle. When they get to the Krebs cycle and you have citrate, but you can't do anything with it. How do you stop making a bunch of citrates? You either have to stop the conversion of pyruvate to acetyl-CoA, or you can just slow down the production of pyruvate. How do we do that? Well, if you slow down PFK. Because if you slow down PFK, you slow down the rest of glycolysis. Right? If you slow down PFK, you slow down the production of ATP, but you also slow down the production of pyruvate. Right? So this is another way that see how these two systems are talking to each other. You have glycolysis, which is anaerobic, but the byproduct of glycolysis, pyruvate, feeds into aerobic metabolism in the Krebs cycle, right? And once that pyruvate is converted into acetyl-CoA, it gets converted into citrate, citrate's aerobic. If citrate's building up, this aerobic metabolite, this aerobic product, can then talk back to the Krebs cycle and inhibit PFK to slow that production down, right? So it's how they all kind of talk to each other. Make sense? Right. So these allosteric inhibitors and activators, ignore fructose 6-phosphate. If we just are looking at these, this says we're using ATP, we're using a lot of energy. This says we already have a lot of it, okay? This says that, you know, we have potentially a lot of substrates to use in aerobic metabolism, but if it's building up, then it's not being used, right? There's something slowing down in that pathway, so we should stop that conversion. Right? We're going to slow down that a little bit. So essentially, the energy state of the cell affects the reaction of PFK. So when there's low energy status, the PFK reaction happens faster. If there's a lot of ATP in the cell, a lot of energy, the reaction happens slower. Okay, so these reactions would happen anyway. It's just that depending on how the environmental statuses of the energy in the cell will make it happen faster or not. Okay? So just to remind you, okay, when we start with glycolysis, we start with glucose, 6-carbon molecule, and then we make glucose 6-phosphate. What enzyme is involved in this step? Hexokinase, right? It uses an ATP. So notice we take a phosphate off of ATP, put it on here. Now we have 6 carbons, 1 phosphate. Next, we turn glucose into fructose. Then we use another ATP. That's the PFK reaction to add another phosphate to this molecule. So we have 6 carbons and 2 phosphates. Now there's 2 arrows here because this 6-carbon molecule will be split in 2. It'll go into a 3-carbon molecule with 1 phosphate and another 3-carbon molecule with another phosphate. So when it splits, then this NAD plus dependent step allows you to add another inorganic phosphate from the cytoplasm to this substrate to make 1,3-biphosphoglycerate. Notice how now we have 2 phosphates on here. Now we can make 2 ATP from this lane and then 2 ATP from this lane. Because if we didn't add this second phosphate, essentially we would just be taking off the phosphates that we put on initially with the start of glycolysis. Right? It takes 2 phosphates for the investment, and then we can produce 4 later in glycolysis. So if we didn't add these other 2 phosphates here in this NAD plus dependent step, we'd essentially just be using ATP to put phosphates on, and then in the second half of glycolysis, we'd just be taking the same phosphates off, right? There'd be no net production positive. But because of this NAD plus dependent step, we can take a inorganic phosphate from the cytoplasm and just add it to that molecule. Kind of like glycogen phosphorylase, right? What's the difference between glycogen phosphorylase and hexokinase? Hexokinase uses ATP to make G6P. Glycogen phosphorylase does not. It just takes a phosphate from the cytoplasm and puts it on that molecule, right? So the net difference is you get 1 more ATP when you start with glycogen because it doesn't cost us an ATP. We're just taking that phosphate from the cytoplasm. This is kind of like a similar concept, right? Here, because we have NAD+, it allows us to take a phosphate from the cytoplasm and put it onto this molecule. Now we've added another phosphate to this molecule, and now the net production of ATP is actually +2 instead of 0, because those two we added from the cytoplasm then become the additional ATP that were produced. Does that make sense? Yes.
Where does NAD plus accept that hydrogen from?
Where does it accept the hydrogen? It's in this, there's an oxidation reaction here. So essentially when it moves the electrons around on this molecule, it takes the hydrogen off, and then in that process it's able to add the phosphate to it, to that substrate. So now the hydrogen that was on this molecule gets taken off and is put onto NADH, and because the hydrogen comes off, the phosphate can go onto that, and that's how that's added here.
Thank you.
Does that make sense? Right. But also remember too, like, there's NADH there, but there's lots of steps in metabolism that require hydrolysis, which, oxidation and reactions, right? So, like, here we're looking at it like, oh, there's NAD plus available here. But you've got to consider, like, there could be, what is the entire state of the cell? Like, how much NAD plus is available overall compared to how much is saturated with hydrogens already, right? Because all these molecules are moving around. We tend to think of it as they're just static, like they're in their lane, right? But all these things are moving around in the cell, especially when you're contracting, things are very mobile, right? So it kind of just depends on what is the overall availability of this chaperone molecule. If there is a lot of NAD+, well then yeah, there's enough to have this oxidation step occur. If there's not enough, there's only a little, well then this is going to happen at a much lower chance, because why? There's less of the substrate for this reaction to occur. Remember the factors that affect enzyme activity: the activators, the substrate availability, the inhibitors, the enzyme concentration. All those factors influence how quickly these reactions occur. So if there's a lot of NAD+, this step here happens very readily, right? If there's not a lot of NAD+ and only NADH, this step is not going to happen readily because there's not enough of this chaperone molecule to accept the hydrogens in this oxidation step. Okay? But having NAD+ available is important because it allows us to add this second phosphate here. Now once you've added this second phosphate, one phosphate comes off to produce an ATP, and then the other phosphate comes off to produce the other ATP, and then we're left with a pyruvate. Cool? So this is what gives us our net +2. Okay, this NAD+ dependent step. Because this NAD+ dependent step is here, we don't actually produce any ATP prior to this step. It's only once this step occurs, then we're able to generate ATP. Okay? And that's pretty much the main point, and that is an NAD dependent step. So just like there has to be a specific substrate available for certain reactions to occur, if this molecule is not here and this step of glycolysis, you will not produce the ATP. Okay, so when we think anaerobically, how are we able to make more ATP? We've got to find a way to make NAD+. One way we can do that is by taking the hydrogens off of NADH and putting them onto something else. That will give us an NAD+. Right, so why do we care about NADH? Because NADH really, it stores energy potential, right? We're not directly making ATP with NADH. We're making ATP when those hydrogens go to the electron transport chain, and then they make ATP, right? So NADH is really valuable because it has a high energy potential, but that's only at the very end of the ETC when we finally get there and make the ATP. Until then, it's just kind of like stored in potential energy. It's not actually energy we can use, right? So ideally, if oxygen is available, then that NADH is reoxidized in the electron transport chain. You make ATP by oxidative phosphorylation. And anaerobic conditions, this NADH is reoxidized by lactate dehydrogenase, which allows us to make NAD+ for those steps in glycolysis. Right? Now NADH is shown in mitochondria where it's oxidized. Right? The NADH actually can be really important because it can be shuttled. Those hydrogens can make ATP later when there's oxygen available. So when we think about like lactate production, high intensity exercise produces lactate, but yes, those hydrogens are on the pyruvate making lactate, but when the exercise intensity goes down, or you slow down, or you go at rest, that lactate can take those hydrogens off of itself, right, with the lactate dehydrogenase, put those back onto NAD+, and then send those to the electron transport chain to make energy, right? So the fact that we're able to make lactate is good because, number one, it allows us to make ATP directly in glycolysis, even if there's not enough oxygen. And then two, all that lactate that's produced is essentially still holding on to those hydrogens, right? So then when you do go into rest and recovery, those hydrogens come off of pyruvate, go back onto NADH, those NADHs go to the electron transport chain, and then we make ATP out of those, right? So it's still good because it still allows us to use those hydrogens in some way, right? We're kind of managing and organizing them while we have this really intense exercise stress, and then once we resolve that, then we can still use those hydrogens later to make the ATP in recovery, right? So the only difference between pyruvate and lactate is lactate has 2 additional hydrogens on it, okay? So in this step, lactate dehydrogenase takes the hydrogens off of NADH, puts them onto pyruvate, and that forms lactate. Once we've taken these hydrogens off of NADH and put them onto pyruvate to make lactate, now we have NAD+, okay? This NAD+ can then be used in glycolysis to make ATP. Does this make sense? So in glycolysis, if you only have NADH, you can't have these phosphates, you can't make ATP. But because LDH can take those hydrogens off and put them onto the pyruvate, then we do have NAD+, and then we can make ATP. Okay? So if we didn't use this process during high intensity exercise, during anaerobic conditions, essentially we would get to a point where we would use all our ATP and then we can't make any more, right? We would have half processed these glycogens, but then we get to this point, we have no way to make NAD+, so therefore we're never going to be able to make ATP. We'd have to wait and stop contracting until there's oxygen supplied, get the NAD+, and then make the ATP. But because we're able to move those hydrogens off of NADH and put them onto pyruvate, that's a way we can make the ATP independent of oxygen, okay? Because we can do that in glycolysis. But we can only do that with carbohydrates in glycolysis. We can't do that with fats, right? Like in the beta oxidation requires NAD+, right? Lipolysis, some steps also require NAD+, right? So lipolysis, beta oxidation, the Krebs cycle, all require NAD+. If you don't have any, none of that substrate can get metabolized in a way to make ATP. But carbohydrates, you do have a way to get that substrate. You do have a way to get the NAD+, right? So that's why that's the only substrate that can be used anaerobically. Following? All right? So, I mentioned that because that concept on your exam, you will be asked, like, why is it so important for NAD+ is available? Like, what's so key about this step in glycolysis? What is the purpose of NAD+ in glycolysis? Why do we care? Also, how it relates to how certain substrates get turned on, how certain enzymes get turned on or turned off. Some enzymes, their activity depends on if NAD+ is available, right? Like, what's the rate-limiting enzyme of the Krebs cycle? Isocitrate dehydrogenase. What's one of the factors that allosterically regulates isocitrate dehydrogenase? NAD+. If NAD+ is there, that enzyme is active. If NAD+ is not there, that enzyme is not active, right? So the Krebs cycle is aerobic, right? NAD+ takes hydrogens, bring them to the aerobic metabolism to make ATP. If you don't have the NAD+, this rate-limiting enzyme, the Krebs cycle, is not active, right? So does that slow down aerobic metabolism? Yes, and if you don't have NAD+, can you make ATP in glycolysis? No, so you can't make ATP with aerobic metabolism because NAD is preventing the rate-limiting enzymes from being active, right? So the Krebs cycle shut down. You can't make ATP here in glycolysis because without the NAD+, you can't produce this ATP down there. But luckily, lactate dehydrogenase will take the hydrogens off of here, put them onto pyruvate, then we do have NAD+ to make ATP, and that's why we can make ATP in high intensity activity. Notice, you only make lactate when you're working out hard enough, right? Like who's ever produced a lot of lactate just going for a light jog, sitting here at your desk? You don't, right? Because in this state, there's not a large need for oxygen, and also your muscles are not contracting, they're not using a lot of ATP, right? So both those factors essentially allow you to use oxygen quite freely to make ATP because it's at rest. But as soon as you start working out really hard and there's not enough delivery of oxygen to the mitochondria, aerobic metabolism starts to back up and slow down. So then that means we can only use glycolysis to make the ATP, and the only way to make the ATP anaerobically, because there's not enough oxygen, is to make lactate. That's why the harder you exercise, the more intense the exercise, the higher the lactates. Because to make more ATP, to do that harder exercise, you had to make more lactate to do that harder exercise, right? The only way to make more ATP to continue to exercise harder is to continue to make lactates. That's why you always see this relationship between really high work outputs anaerobically and really high lactate concentrations. Right? Is that clear? So understanding that relationship between how lactate's produced, why it's produced, how that feeds into those two anaerobic and aerobic kind of pathways is important, because on the exam you will get questions about like which metabolic pathway is most likely being used in this type of activity, or this duration of activity, or this intensity of activity. And if you're not really clear like what is high intensity exercise and what's the substrates used or the pathways, you might be saying one pathway or substrate that's totally not relevant to what the real answer is, right? So you've got to see kind of how these pathways work together, and why if oxygen is present, we're using this pathway. If it's not present, we're using these pathways, okay? I think I mentioned that in the first class, right? Exercise physiology, all it boils down to is oxygen present or not? And based on that question alone, the rest of the metabolic pathways will all kind of modulate and change and readjust to try to maintain some level of homeostasis, if it's there or if it's not, okay? So, remember that when we make glucose 6-phosphate, right, this is the starting molecule of glycolysis. When we break down glycogen, it makes a glucose 1-phosphate. Notice how like these phosphates are on different locations, right? It's on this carbon up here, it's on this carbon down here, right? So yes, it's the same number of carbons, the same number of phosphates, but different location. Enzymes work with molecules that have specific structures. If it's not in that structure, it's not going to work with it, okay? So when we have glucose 6-phosphate, okay, we can use it during glycolysis, okay, and that eventually produces pyruvate. In the liver, we also have liver glycogen, right? That liver glycogen can make glucose 6-phosphate, but in the liver, once we make glucose 6-phosphate, we actually do have an enzyme. It's called a glucose 6-phosphatase, right? A phosphatase takes a phosphate off. You have to know that kinases add phosphates to things. Phosphatases take phosphates off. So because the liver has glucose 6-phosphatase, it can take that phosphate off of glucose, and now it can leave the liver, and now it can increase circulating blood glucose levels, right? So that's why the liver does that, and skeletal muscle cannot. Now, when you think about glycogen metabolism, glycogen phosphorylase is also subject to allosteric regulation by those same molecules of ATP breakdown, right? So AMP, ADP, ATP, glucose 6-phosphate, all those factors regulate whether glycogen phosphorylase will be highly active or inhibited, right?
Doesn't calcium have an effect on glycogen phosphorylase?
It does, yeah, look at that in exercise, it does, yeah. So, AMP and ADP activate phosphorylase. Okay. So, what's the purpose of glycolysis? What's the primary purpose of glycolysis? To make ATP, right? It uses the glycogen, breaks it down to glucose, all these steps, producing ATP. So the primary goal is PFK will be active if there's a need for the ATP, right? So we can see that if we're using ATP, just like the effect on PFK, we're making more AMP, we're making more ADP. Those two factors increase the activity of phosphorylase activity, okay? On the contrary, if we already have a lot of ATP or have a lot of glucose 6-phosphate, that will actually inhibit phosphorylase activity, okay? Yes?
So, does like the ATP or the G6P have to bind to the enzyme to inhibit it?
They can bind to the active sites, right? So essentially it's like, like musical chairs, right? So you only have so many seats, right? And if somebody else sits in your seat, well, I guess you're out of luck, right? You can't sit there. That's partly what happens in some cases that ATP, like for this example, ATP can sit into the active site where AMP is supposed to bind. And so if ATP's in there and not AMP, phosphorylase can't be active, right? But if you're using the ATP, it gets broken down, now the active site's available, then AMP can sit in the active site and that will increase the activity of phosphorylase. So they can kind of sit into the active sites and sort of like interfere with the correct substrate from activating and sitting where it's supposed to. Sometimes these molecules can actually interact with other areas of the enzyme and change its structure in other places. So then, like, maybe the active site is available, but because there's another activator or inhibitor somewhere else on the enzyme that's changed the structure, now the active site kind of sits like this. So it's still there, but it's not in a formation that the actual substrate can sit in and activate it fully, right? So there's other things that modify the structure of these enzymes and proteins that can inhibit or increase its activity without actually even sitting in the active site. It's just changing the structure of the molecule. You also have other things that do sit in the active site and either stimulate it or inhibit it as well. So all these factors is what kind of fine-tunes how active these enzymes are to facilitate metabolism to make more ATP. So good question. So yeah, so when we think about glucose 6-phosphate, remember if there's a large need for ATP, whenever we make glucose 6-phosphate, it's going to get pushed through glycolysis as soon as it can to make ATP. If glucose 6-phosphate is building up, it means that either one, there's not a big need to make ATP because certain molecules in glycolysis have slowed down, right? So that if, like for example, if PFK is not highly active, is it going to readily take fructose 1-phosphate into fructose 1,6-phosphate? No. If PFK activity goes down, then we're not converting fructose 1 into fructose 1,6. So therefore, fructose 1 stays high. And if fructose 1 stays high, that starts backing up the rest of the chain. So then there's a lot of G6P. A lot of G6P, why do we need to keep breaking down glycolysis if we're making more G6P, right? Let's stop making G6P because we already have enough of it. Okay, so these work in opposition when if glycogen phosphorylase breaks down glycogen so we can make more ATP, when you have a lot of ATP or have a lot of G6P, that will inhibit phosphorylase activity because it's telling that enzyme essentially we already have enough energy. We don't need to keep breaking down more. You have more glycogen in your total body muscle, your total body glycogen in muscles is more than what you have in your liver, right? Obviously, how much glycogen that's in the muscle depends on the size of the muscle, right? So I'm not saying like the muscle has more glycogen than the liver, but as a function of your total body glycogen stores, there's more glycogen in your entire body versus singularly in your liver, right? So we wouldn't want the muscle to just expend all the glycogen, because essentially once you run out of that glycogen, we can't resynthesize it unless we have carbohydrates. So we have to be able to stop breaking down glycogen very quickly if there's not a need for it, okay? We'll learn in a few chapters, epinephrine, adrenaline, right? It accelerates glycogen breakdown, okay? It increases glycogen breakdown, but as soon as we don't need to keep breaking glycogen down anymore, the epinephrine receptor is turned off so that it stops that process without overconsumption of glycogen, okay? Epinephrine is a very powerful stimulator of substrate mobilization. So imagine if you just had epinephrine's released, interaction with the receptor, and then it just breaks down all the substrates in your muscle, right? Like, what if someone just comes in the room and scares you, right? Huge epinephrine response. Would we want all our substrates to be broken down because someone's scared? No, we want it when we need it to run from a bear, right? Or, you know, or Texas Chainsaw, or whatever. Like, I don't know, you know. I watched a scary movie this weekend, so it's like, anyway. So the idea is that we want to be able to regulate and stop substrates from being used too quickly if it's not the right need for it, right? So our muscles do that. Our muscles will actually stop the epinephrine receptor from breaking down more substrates than it needs to, so that we still have the substrate if the time came for it, okay? So, glycogen synthase is allosterically activated by glucose 6-phosphate. So, if phosphorylase breaks down glycogen, what do you think glycogen synthase does? Makes it synthesizes it, right? The function of these enzymes is often in the name. Glycogen synthase synthesizes glycogen. It stores glycogen. It makes glycogen. So it's actually no wonder, if we think about it, if glycogen synthase inhibit, or if glucose 6-phosphate and ATP inhibit phosphorylase, then glucose 6-phosphate will stimulate glycogen synthase, right? Because if G6P is stopping glycogen breakdown, then actually that G6P should tell the enzyme synthase to be active, so we can store that G6P as glycogen when we need it. Okay, so they work in opposition. You're never really going to see in glycolysis, you're not going to see in metabolism as a whole, you're not going to see catabolic reactions and anabolic actions happening maximally at the same time. Okay? So you're not going to see you're breaking down glycogen and also storing glycogen at the same time. Because you either need to use that glycogen for energy to make ATP, or you don't need to make more ATP. And so then we'll take that glycogen and store it as, we take the glucose and store it as glycogen, okay? It's almost like this, you see like at a construction site, you know, someone's taking the brick and they're, you know, building, and someone else takes that brick and puts it back on the pile, so the building never gets built because the person taking the brick is thinking they're doing the work, and then the other person taking the brick and put it back on the pile, right there. So one's breaking things down, the other's trying to build it up at the same time, nothing gets done, okay? In metabolism, it's the same way. Glycogen breakdown, we're either breaking it down or we're storing it, not at the same time. Lipids, either lipolysis is happening, we're making energy from fats, or we're storing those lipids as energy. Protein synthesis, either the muscle is contracting and it's breaking down those tissues, breaking the tissues apart, or we're in rest and recovery and then we're resynthesizing those amino acids into a new protein. We're not going to be breaking down protein and remaking it at the same time. It just doesn't work like that. Because the signals interfere with each other. They prevent, if this one's highly active, that same signal actually tells the other one to stay off. Is that clear? Okay, why do I say that? Because you could get questions on the exam about certain types of intensity of exercise, you know, maybe certain training statuses, things that I'm hypothesizing there are examples on the exam. And you have to know, like, well, if synthase is doing this and it's during exercise, I know that synthase has to be off, because why would we synthesize glycogen during high intensity exercise? that wouldn't be the answer. The answer has to be phosphorylase, right? So I will give you examples about the enzymes and hormones and exercise intensities and situations, and you've got to be able to think like, oh, like, what does this mean, right? If synthase is storing glycogen and we're doing exercise, that doesn't make sense that would be active right now, right? You got to kind of put those together. So calcium, let me ask you this, when is calcium released in the cell? When there's an action potential, right? So when a motor unit fires, that action potential goes down the motor axon, goes to the terminal end plate, electrical, chemical, electrical, propagation down sarcolemma, down T tubules, release from the SR. Okay? So when there is voluntary contraction, there is the release of calcium in the cell. The release of that calcium, how does that allow muscle to contract? Right? The actin myosin can bind. When actin and myosin bind, how do they produce force? The power stroke, right? How does a power stroke happen? ATP breakdown, right? On the head of myosin, there's a myosin ATPase that splits ATP into ADP, inorganic phosphate. Once that hydrolysis happens, the myosin head is loaded, ready for the power stroke, position, shortening, release, reload, right? So when calcium is present in the cell, the muscles can undergo contraction. If they're contracting, they're using ATP. So the release of calcium essentially can prime glycolysis to start making more G6Ps, because the idea is if calcium is released, the muscle's probably contracting. If it's contracting, it's using ATP. How can we make more of it? Oh, let's turn on phosphorylase to start breaking down some glycogen so that during that contraction we can make ATP a little easier or a little faster. Right? So calcium breakdown, or calcium helps regulate glycogen breakdown. The release of calcium activates phosphorylase so that we can break down more glycogen, so we have more energy, more substrates available for use during exercise. Yes?
Will calcium inhibit synthase?
I think, off the top of my head, I don't think calcium directly inhibits synthase, but I'd have to double check for you on that. But I think overall the other factors related to exercise, like ANP, the breakdown of ATP, all those other things are more likely to inhibit synthase activity more so than the calcium itself. The calcium for sure stimulates glycogen phosphorylase, but I think it's the other factors that actually inhibit the synthase activity. So, if you look here, there are a lot of charts and stuff on this in this class, but if you look up at this corner pocket, this says calcium concentration. The calcium concentration increases. Notice how if we follow this arrow, it turns on phosphorylase kinase. Phosphorylase kinase can turn on glycogen phosphorylase. Right? So the release or the increase in calcium in the cytoplasm turns on glycogen phosphorylase. Also notice, up here we have the concentration of ATP. If the concentration of ATP decreases, that means AMP is increasing, and that stimulates glycogen phosphorylase. Right, so both of these factors are stimulating glycogen breakdown. Why? Because they're acting on a common enzyme that regulates breakdown. Up here we have adenylate cyclase. Right? Adenylate cyclase is an enzyme inside of or close to the cell membrane. It's activated by adrenaline or epinephrine. Right? Epinephrine binds to its receptor. The receptor increases the activity of adenylate cyclase. Adenylate cyclase turns on CAMP, pKa, phosphorylase kinase, glycogen phosphorylase. So what's good here is that whether it's coming from the nervous system, action potentials, calcium release, or it's coming from the energy use, ATP is going down, AMP is going up, or physiological stress, the release of epinephrine stimulates the receptor, turns on pKa, then turns on phosphorylase. All three of those factors are stimulating glycogen phosphorylase to turn on, so that whatever the stimuli is, we're going to be able to access that glycogen very quickly so we can make the ATP quickly when needed. Imagine if the only way for us to break down glycogen was if enough ATP decreased in the cell. or if we had to make sure there had to be an epinephrine response. Right? Like I'm doing bicep curls. I need more glycogen. I can't get to it because I don't have a systemic epinephrine response. I guess I gotta stop contracting. But because calcium release also stimulates phosphorylase, I can access that glycogen locally in the muscle without needing a systemic response. And actually, would I want a systemic epinephrine response if I'm only contracting one muscle? No, right? So, and actually the fact that we can have very localized regulators of glycogen metabolism and also have systemic regulators as well is actually really beneficial because we're not relying on just one modality to access substrates when we need it. Does that make sense? So yeah, think about the in big picture kind of thing. But let's go back to exercise intensity. Higher intensity of exercise, more epinephrine responses, more glycogen breakdown. Higher intensity of exercise, more type 2 fiber use. Type 2s are better at handling calcium, more calcium is released, more glycogen breakdown. Don't you use more carbs during high intensity exercise? Yeah, one, because we only use that fiber type. We primarily use that fiber type, type 2s, which primarily use carbs. And the epinephrine release stimulates more carb breakdown, right? More calcium is released because more firing, more calcium in those fibers, more glycogen breakdown, more carb use, right? So all these factors are related. So to reiterate, the relationship between glycogen, phosphorylase, and synthase activity are inverse, right? When phosphorylase activity is very high, synthase activity is very low. When synthase activity is high, phosphorylase activity is low, right? So when we need energy, our muscle's going to try to make the energy. If we're adding energy exogenously, so we're adding glucose, right? Phosphorylase activity goes down, and synthase actually takes that glucose and synthesizes G6P and then synthesizes glycogen. Okay? They're working in opposition. You'll never see these on at max intensity at the same time because they have two opposite functions. So last things to remember here, right, it kind of reiterates those circled parts of glycolysis. Hexokinase is important. It's the enzyme that converts glucose into G6P. It uses an ATP. Glycogen phosphorylase is important. It's an enzyme that works specifically with glycogen breakdown. Again, I'll reiterate, do not confuse these on the exam. If you confuse them, you'll get multiple questions wrong on the same topic. So please don't confuse them. In fact, the name for one of them is right in the enzyme name. Glycogen works with glycogen phosphorylase. Hexokinase works with the sugar, with the glucose, okay? Now that glycogen phosphorylase is important because it does not use an ATP to phosphorylate glucose. That's where the net difference in ATP production comes from when you start with glucose versus the glycogen, because there is not an additional use of an ATP with phosphorylase. Where does that phosphate come from?
Cytoplasm.
Cytoplasm, right? That phosphate is added to the glucose to make G1P. It comes from the cytoplasm. Fructose, PFK, fructose phosphofructokinase, right? This step is important because it uses another ATP. It's highly regulated. It's a rate limiting enzyme, and it's irreversible. Right? It uses an ATP to make fructose 1,6 bisphosphate. Okay. This step here, phosphoglycerate kinase, essentially this is the first ATP we make. Notice we take a phosphate off of 1,3-diphosphoglycerate. That one phosphate makes an ATP. Now we just have a 3-phosphoglycerate. A few more steps. Pyruvate kinase takes the phosphate off of phosphoenolpyruvate and makes pyruvate. We've also produced an ATP. Right? So these are the steps where we're using an ATP. We're not using ATP, but it's a priming reaction. We're using an ATP here. We make an ATP. We make an ATP. And lastly, this step is important. Lactate dehydrogenase. It's the enzyme that's used to convert pyruvate into a lactate. How does it do it? It takes the hydrogens off of NADH, puts them onto pyruvate to synthesize lactate. Now remember, on the exam, I will ask you questions. Which substrates and enzymes are necessary for this reaction to occur? So if I said, lactate dehydrogenase, right? What do you need for that reaction to occur? You need the enzyme. You need the NADH, right? And you need pyruvate, okay? If you just said pyruvate and LDH, that's wrong, because you still need the hydrogens, right? If you just said the NADH and pyruvate, no, you need the enzyme as well, right? So make sure you take note of these things when on the exams, because some of them are the right answer because they have all the components there. Other answers might have some right parts of it, but the other parts are either incomplete or the wrong other substrate, okay? That's pretty much that, okay? We will cover glycolysis and more glycogen metabolism when we talk about the hormonal responses in next week, actually. All right, now we're going to aerobic metabolism.